Welcome, curious minds, to The Deep Dive. You've sent us an incredible stack of sources, really fascinating stuff. And today we're embarking on, well, a really crucial journey. It sits right at the heart of understanding our biology and maybe more importantly, how we approach disease.
Mhm.
This deep dive is pretty foundational for anyone immersed in genomic medicine, and we're about to explore some truly compelling territory. When we talk about genomic medicine, often the first thing that probably springs to mind is just analyzing the DNA sequence itself.
Yeah.
We tend to think of our genes as these fixed blueprints, dictating everything. But what if there's a whole other dimension, a really dynamic layer that dictates how those genes actually behave, whether they're read, how much they're read, or even if they're read at all? A layer that's constantly being rewritten in response to our lives, our environment, even our experiences.
That's precisely it. That's the core idea. Today, we're plunging into the world of chromatin remodeling and organization. It's really not just about the linear sequence of our DNA, the A's, T's, C's, and G's. It's about how that DNA is meticulously packaged and, crucially, dynamically managed within every single cell in our body. You can almost think of it as the grand orchestrator. The orchestrator behind how our single identical genome can somehow give rise to over 400 vastly different cell types and tissues throughout our body. Each of these cells, whether it's a neuron or a skin cell or maybe a liver cell, might have the same underlying genetic blueprint, the same genes, but it possesses its own unique epigenome. It truly is the instruction manual, telling each cell how to read its particular genetic blueprint. Not just what's there, but how to use it.
Okay, so our mission for this deep dive then is to unpack these intricate mechanisms of chromatin. We need to start from its fundamental units, the basics, and go all the way up to its stunning, quite complex 3D architecture. We're going to try and reveal how these dynamic changes influence, well, pretty much everything, from the initial formation and differentiation of a cell type to its ongoing function, and critically, how they play a role in disease, including opening up some really exciting new avenues for therapies. We'll be drawing deep insights from the resources you've provided us, including the very illuminating Molecular Medicine, How Science Works by Karlberg, Valoyer, and Molnar, which lays out much of this foundational understanding really well. So why is this particular deep dive so important for you? Especially if you're engaged in genomic medicine, why focus here?
Well, understanding chromatin's dynamic dance is, I think, truly a shortcut to comprehending not just how our bodies function, adapt, and respond to the world around us, but also, unfortunately, how they sometimes develop complex diseases like cancer or metabolic syndrome or various immune disorders.
It's where a lot of that missing heritability in these complex diseases seems to lie. It moves us beyond a purely inherited genetic sequence to a much richer, more dynamic picture, and ultimately maybe a more treatable picture of health and disease. This is where, the real action happens beyond the simple A, T, C, G sequence. Let's really unpack this.
Absolutely. Let's begin with that big picture concept you mentioned. Every cell in our body, with very few exceptions, essentially contains the same core genome. That fundamental DNA sequence is the same. Yet what's truly amazing, and maybe a bit mind-bending at first, is that this single genome can give rise to hundreds of vastly different epigenomes. This is the incredible molecular mechanism that ensures, say, a kidney cell remains a kidney cell. It prevents it from inexplicably transforming into maybe a liver cell. It's all about maintaining that cellular identity and specialization throughout our lives, right from the moment we begin as just a single cell. Consider the incredible journey of embryogenesis. Embryonal stem cells, or ES cells, they start as these highly plastic, undifferentiated cells. They hold the potential to become basically any cell type. As the embryo develops, these ES cells undergo differentiation, giving rise to all the various specialized cell types and tissues that form a complete organism: muscle, nerve, skin, everything. And it's not just during development. Even in adults, our bodies continuously replace lost cells. Think about those in our bone marrow, our skin, or our intestinal lining. This happens through the ongoing differentiation of adult stem cells. Now, what's critical to understand here is that the fundamental underlying mechanism for all these intricate differentiation processes, from the very first divisions of an embryo to the continuous renewal in an adult, is the epigenetic programming of chromatin structure. It's like the cell receives a set of instructions, not just about what genes are present, but more importantly, when and how much to express them. And these instructions become stable, mitotically heritable over time, locking in a cell's identity.
That's a powerful idea, the stability of cellular identity being actively maintained. So it's not just about building the body initially, but keeping it running in a healthy, ordered state. And this brings us to a really critical point: how these intricate epigenetic instructions can sometimes go wrong, how they can go awry and profoundly influence disease. Yeah, our sources make it abundantly clear epigenetics and immunity are, well, truly fundamental to understanding human biology across the entire lifespan. It's not a niche topic. This starts right from the earliest moments of embryogenesis, continues through the daily maintenance of our bodies, what we call homeostasis, and it extends all the way into the complexities of aging. What's even more significant, and maybe often overlooked, is that many common diseases we grapple with today, think cancer, metabolic syndrome, various immune disorders that have both a genetic an inherited component and a dynamic epigenetic component. They are truly intertwined. You can't really separate them. Let's focus on cancer for just a moment because it's such a prime and striking example of this interplay. For a long time, cancer has primarily been viewed as a genomic disease, right? One caused by accumulated DNA mutations, things like point mutations, translocations, deletions, or amplifications in the DNA sequence itself. But what the cutting edge research, like what's in our sources, reveals is that tumorigenesis, the actual development of a tumor, also profoundly involves abnormalities in cellular identity. Cells forget what they are, it involves altered immune responses and major fundamental changes in the cell's transcriptome, the full set of RNA molecules expressed. All of these crucial changes are rooted in alterations to our epigenome. The way genes are regulated is broken. In fact, most types of cancer don't just carry mutations in their genome, they carry widespread mutations and dysregulations in their epigenome as well. This immediately raises a fascinating question. If epigenetics is so dynamic and absolutely crucial for normal cellular function and identity, what precisely happens when this intricate system goes wrong? This shift in understanding fundamentally moves us, I think, from a purely deterministic genetic view of disease to one where the environment, our lifestyle choices, even just aging, can play a profound role by altering our epigenome. It reveals much of that previously missing heritability in complex diseases. It's a real game changer in how we think about disease origins.
It truly is. And to understand how this can go wrong, we first need to appreciate how DNA is organized in the first place.
Yeah.
It's not just a random mess. Our DNA isn't just a loose string floating randomly inside the nucleus, it's precisely packaged. This packaging is the very first level of chromatin organization, and it's absolutely essential. You have meters of DNA in almost every cell. It's astonishingly long, and it has to fit into a microscopic nucleus. Imagine trying to cram miles and miles of thread into a tiny thimble. The packaging has to be incredibly efficient. So at its most basic level, DNA is meticulously wrapped around these proteins called histones, like thread around a spool. These histone DNA complexes form fundamental units known as nucleosomes.
Right, the beads on a string analogy we often hear.
Exactly, that's a great visual. The string is the DNA, and the beads are these nucleosomes. What's crucial, though, is that these nucleosomes, these beads, are themselves further organized and compacted into even higher order structures. Together, all of this makes up what we call chromatin. Now, this packaging isn't just about compaction, about fitting it all in. It fundamentally determines whether genes are accessible or not. It's about regulation. If DNA is tightly wound around these histones and further compacted into these higher order structures, then the essential cellular machinery, things like transcription factors and other regulatory proteins, they simply can't reach the genes to turn them on. They can't physically get access. So this accessibility, or the lack of it, is where chromatin organization becomes absolutely key in dictating gene regulation. It's the first gatekeeper, you could say.
Okay, so if I'm understanding this correctly, it means some parts of our DNA are readily accessible, almost like an open book, easy for the cell to read and act upon, while other parts are really tucked away and hidden, almost like they're locked in a vault, inaccessible.
That's a perfect way to put it. An open book versus a locked vault. During interphase, which is that long period when a cell isn't actively dividing, but is busy doing its job, performing its functions, a remarkable, maybe even surprising, 90% of the genomic DNA in terminally differentiated cells. So cells that have committed to a specific function, like a muscle cell or a nerve cell, 90% of their DNA is actually inaccessible to transcription factors.
90%, that's huge.
It is huge. This tightly packed, silent form of chromatin is aptly called heterochromatin. And it's effectively the cell's default state for genes it doesn't need to express right now, or perhaps ever in that cell type. In stark contrast, the accessible, active form of chromatin where genes can be readily read and expressed is known as euchromatin.
Okay, heterochromatin is the closed vault, euchromatin is the open book.
Exactly. Think of heterochromatin as that tightly rolled up scroll, maybe secured with a ribbon and put away on a high shelf, impossible to read. Euchromatin. That's the scroll unrolled, spread out on a table, ready for immediate reading and interpretation by the cell's machinery. It's a beautifully elegant, yet profoundly impactful system for managing our vast genetic information.
And the organization gets even more complex, right? It goes far beyond just being tightly or loosely packed. The 3D organization within the nucleus, how it all folds up, is also critical.
Absolutely. It's not just linear packing. Chromosomes aren't just randomly arranged strands of spaghetti jumbled in the nucleus. They adopt very specific, highly non-random organizations in three-dimensional space. This architecture directly impacts gene function. What's truly fascinating here, and something researchers are really digging into now, is how DNA looping allows physically distant genomic regions that might be far apart on the linear chromosome, to actually contact each other in this 3D space. These aren't arbitrary connections. They form what we call architectural and regulatory loops, often stabilized by specific proteins that act like molecular anchors, holding them together. And here's a critical insight from the research. Most of these architectural loops seem to be identical to what scientists call topologically associated domains, or TADs.
The TADs, right. I've seen that term. Topologically associated domains.
Yes. These TADs are fundamental units of chromosomal organization. They essentially segregate our vast genome into at least 2,000 distinct domains, maybe more. Each of these domains typically contains groups of genes that are co-regulated, meaning they tend to be switched on or off together, acting as a functional unit.
Yeah.
Now, defining the boundaries of these TADs are often special stretches of genomic DNA called insulators. These are bound by specific proteins, and a key example, a really important one, is called CTCF.
CTCF.
These insulators, bound by proteins like CTCF, act like molecular fences, separating functionally distinct genomic regions. So you can imagine a map of our genome. One neighborhood, or TAD, might be a quiet residential area with silent genes, mostly in that heterochromatic state. The very next neighborhood, right next door, could be a bustling commercial district with actively transcribing genes, largely in the euchromatic state. The CTCF proteins are essentially the molecular architects, building the fences, defining these boundaries, and ensuring proper functional segregation. Things in one TAD don't inappropriately influence things in the next TAD. This immediately raises a profound question, doesn't it? How do cells know precisely where to put these fences? How are they maintained? And perhaps even more fascinatingly, how do they dynamically change or adjust them when a cell needs to shift its identity or adapt its function, say during development or in response to a signal?
That's a truly profound question because it really hammers home that this isn't some static blueprint just sitting there. It's constantly being adjusted, constantly in this dynamic, intricate dance. So what are the actual molecular tools then? The key players that make these incredible and precise changes possible? Who's doing the adjusting?
Right. So when we talk about how chromatin's accessibility is controlled, how these changes happen, we're delving into a world of dynamic chemical modifications. These happen to both the histone proteins, the spools the DNA is wrapped around, and sometimes to the DNA itself. These modifications are like temporary instruction tags, post-it notes, if you will. They're introduced by specific enzymes that we aptly call writers. They write the instructions onto the chromatin.
Writers, okay.
Then these marks are interpreted or read by other proteins, logically known as readers. They recognize and bind to these specific modifications, triggering downstream cellular responses like recruiting other proteins or changing the chromatin structure.
Makes sense. Writers write, readers read.
Exactly. And just as easily as they can be added, these marks can also be removed by another set of enzymes called erasers. They wipe the slate clean or change the instruction. The intricate, constant interplay between these nuclear proteins, the writers, the readers, and the erasers is absolutely essential for finally controlling gene expression. It ensures that the right genes are turned on or off at the right time in the right cell. It's a constant cycle of writing, reading, and erasing these epigenetic marks.
Okay, so let's talk about some specifics. One of the most crucial and maybe best studied modifications is DNA methylation, right? Adding something directly to the DNA.
Yes, DNA methylation is a cornerstone of epigenetics. This process involves the addition of a small chemical group, a methyl group, CH3, directly to cytosine bases within the DNA strand itself. This primarily occurs at what we call CpG sites. These are regions where a cytosine nucleotide is immediately followed by a guanine nucleotide in the DNA sequence.
CpG sites.
Got it. Now, the writers of these methylation marks are a family of enzymes called DNA methyltransferases, or DNMTs. Specifically, DNMT1 is known as the maintenance methyltransferase. Its main job is to copy existing methylation patterns onto the new DNA strand during replication, ensuring that this epigenetic memory is passed down to daughter cells when a cell divides. Then you have DNMT3A and DNMT3B. These are the de novo methyltransferases. They establish new methylation patterns, often during development or in response to environmental signals.
So one maintains, the others create new patterns.
Exactly. But just as easily as these marks are added, they can also be removed or modified. The erasers in this system are the 1011 translocation enzymes, picky enzymes, TET1, TET2, and TET3. Interestingly, they don't directly remove the methyl group. Instead, they oxidize the 5-methylcytosine, the methylated base of 5-PC, to 5-hydroxymethylcytosine, 5-HAMC, and then potentially to further derivatives. This oxidative pathway eventually leads to active DNA demethylation, removing the mark. And then, of course, there are specialized reader proteins that recognize these modifications. Some bind specifically to unmethylated cytosines, others bind to 5-MeLa-C, triggering different downstream effects on gene expression. Now, how does methylation actually work? It directly modulates the DNA binding affinity of transcription factors. It changes how proteins see the DNA. What's fascinating is the nuance here. Some transcription factors bind better to methylated DNA, some bind worse, and some are completely unaffected.
So it's not just simple silencing?
Not at all. It offers a powerful and incredibly nuanced form of gene silencing, or sometimes even activation, depending on the context, ranging from flexible, reversible repression to a highly stable, almost permanent inactive state. A classic example we mentioned earlier is CTCF, that critical genome regulator with 11 zinc finger domains that helps define TAD boundaries. Its binding to DNA is negatively influenced by DNA methylation at its binding sites. This is particularly important in genomic imprinting.
Yes, imprinting.
Which controls the monoallelic expression of over 100 genes, meaning only one copy, either the maternal or paternal gene, is expressed, and methylation is key to silencing the other copy. So, a simple methyl group addition can completely change how a crucial protein like CTCF interacts with our DNA, fundamentally altering gene activity across large domains.
It's truly mind-boggling, these layers upon layers of regulation. Okay, so beyond the DNA itself, the very histone proteins, the schools, are also extensively modified. This is often referred to as the histone code, right? Suggesting a complex language being written and interpreted there too.
That's right. The histone code is a concept suggesting that combinations of modifications on the histone tails act like a code, read by the cell to determine gene expression states. And histones undergo a vast array of these post-translational modifications. The most studied are probably methylation and acetylation, particularly on lysine residues in the histone tails. But there's also phosphorylation, ubiquitination, chromoylation, and many others. It's incredibly complex. These modifications aren't just passive decorations, they directly affect the chromatin's density and accessibility. For example, acetylation generally tends to neutralize the positive charge on lysines, loosening the interaction between histones and the negatively charged DNA, thereby opening up the chromatin and promoting gene expression.
Acetylation equals active, usually.
Generally speaking, yes. Conversely, some methylation marks can lead to compaction. Critically, these modifications also serve as precise binding sites, docking platforms, for those effector proteins we talked about, the readers. This includes sophisticated chromatin-modifying complexes and chromatin remodeling complexes that physically reposition nucleosomes. Ultimately, these histone modifications profoundly modulate both the initiation and the elongation phases of transcription. They have a direct and significant impact on which genes are expressed and to what extent, thereby shaping the entire transcriptome, the complete set of RNA molecules in a cell at any given time.
Okay, let's delve into some specific marks and what they signify, because this is where the code really starts to reveal its meaning. The sources mentioned several key ones.
Yes, there are some classic examples. For instance, H3K4ME3, that's trimethylation of lysine 4 on histone H3, is found specifically and consistently right at active promoters. It's a very strong signal, basically a green light saying this gene is active or poised for action. In stark contrast, H3K27E3 trimethylation of lysine 27 on histone H3 is strongly correlated with gene repression, often spanning much larger genomic regions, silencing entire gene clusters. This mark is laid down by the polycomb complex, which is crucial for developmental gene silencing.
So K4-trimethyl is active, K27-trimethyl is repressive.
Generally, yes. And then you have marks like H3K93 and H4K2083. These are typically enriched near the boundaries of large heterochromatic domains, the silent regions we discussed. They act almost like signposts indicating keep out or reinforcing those silent states. But here's where it gets really interesting and complex and highlights the nuance beyond simple on-off switches. H3K4ME3, the activating mark, and H3K27E3, the repressive mark, can actually coexist at the same gene promoter in what are called bivalent domains.
Bivalent?
Yeah.
Both active and repressive marks at the same time. How does that work?
It sounds contradictory, but it's incredibly important, especially in embryonal stem cells and during differentiation. These bivalent domains keep genes in a sort of poised state. They provide the potential for both rapid transcriptional activation and repression. Think of it like a car with one foot on the gas, H3K4ME3, and one foot on the brake, H3K27E3. The gene is held in check, but ready to go quickly in either direction, activate or stably repress, depending on the specific differentiation signal the cell receives. And as you might expect, when these bivalent domains are dysregulated, when that delicate balance is thrown off, it can indeed lead to various diseases, including developmental disorders and many types of cancer, where cells lose their proper identity.
Wow, so it's not just on or off, but poised for action too.
Precisely. So you can really think of histone modifications as this incredibly sophisticated system of sticky notes or flags on the DNA packaging. Each combination carries a precise instruction for the cellular machinery. Open here, close there, stay poised, recruit this protein. It's a highly sophisticated, yet elegantly simple way to control the flow of genetic information that goes far, far beyond just the DNA sequence itself. So with all these different marks and all these players, the writers, readers, and erasers, how do all these diverse signals and modifications actually come together? How do they integrate to truly orchestrate cellular function? It can't just be a random collection of tags. There must be some higher level coordination. That's a crucial question because it really highlights the integration aspect. It's not just individual marks. It's the combination and the context. What's truly remarkable is that signals deriving from various signal transduction pathways, many of which begin right at the cell surface, responding to external cues like hormones or growth factors, are integrated directly on chromatin. This often happens through modifications at those histone tails we just discussed. Specific enzymes, the writers and erasers, are often downstream targets of these signaling pathways, so an external signal can directly lead to a change in histone marks at specific genes. This creates an incredibly intelligent system where multiple inputs occurring over time can actually be stored or compounded directly on the chromatin itself. It builds up a history of cellular experience, modulating gene expression accordingly.
Like a memory trace on the chromatin.
In a way, yes. For instance, specific writers like histone acetyltransferases, HATs, and lysine methyltransferases, KMTs, add these marks, often in response to activating signals. Conversely, erasers such as histone deacetylases, HDACs, and lysine demethylases, KDMs, remove them, responding to different, often repressive signals. Then, a diverse array of reader proteins equipped with specialized domains like chromodomains, PhD fingers, and bromodomains recognize these specific modified sites. This recognition then triggers downstream effects, ultimately influencing whether a gene is expressed or silenced. Now, connecting transcription factors, which bind to enhancers often far away, to the actual gene promoter involves hundreds of co-activator proteins. They bridge this gap. A key player in this massive signal integration mentioned in our sources is the multi-subunit MED complex, also known as the mediator complex.
The mediator complex, okay.
This is an enormous protein complex, really a molecular machine. It physically interacts with activated transcription factors bound at enhancers and also directly with RNA polymerase II, the enzyme responsible for actually transcribing genes into RNA at the promoter. This suggests that gene regulatory signals from a multitude of different sources, external stimuli, internal metabolic states, developmental cues, are all funneled through and processed by the MED complex. It acts like a central processing unit, integrating all these inputs and delivering a properly calibrated output signal to the basal transcriptional machinery, ensuring precise and appropriate gene expression levels.
So it takes all the inputs and decides the final output level for the gene.
That seems to be its major role, yes. This intricate network is so complex and interconnected that it naturally raises an important question. How does this incredibly sophisticated, integrated signaling network precisely contribute to a cell's ultimate fate, its stable identity, and its ability to respond appropriately and adapt to its ever-changing environment? That's really the crux of much ongoing research.
That idea of chromatin acting as a sort of information hub, integrating signals and storing history, is truly powerful. It paints a much more dynamic picture of the genome. And the sheer dynamism of the epigenome, this constant reprogramming and fine-tuning, is perhaps most dramatically evident during development, the very sculpting of an entire organism from just a single cell. It's truly a marvel to consider the precision involved.
Absolutely. Development is where we see the most profound epigenetic reprogramming. Our sources highlight that during pre-implantation development, right after fertilization, there's a massive genomic epigenetic reprogramming event. This essentially resets the zygotic epigenome, the epigenome of the newly formed embryo, for what we call naive pluripotency.
Like wiping the slate clean?
Exactly like wiping the slate clean. The cells of the very early embryo are stripped of most of their inherited epigenetic marks, primarily DNA methylation, making them ready to become any cell type. This allows for totipotency and pluripotency. This initial, profound reset is far more extensive than almost any epigenetic changes that happen later in life. It's a fundamental reprogramming. What's truly fascinating is the hierarchy of events, the timing. DNA methylation marks, which tend to be quite stable and serve to lock in cell identity later on, are typically added or removed after initial changes in histone modifications have occurred. They often represent the final, stabilizing step at the end of a differentiation process, ensuring that a cell's identity, once established, is durably maintained and passed on through cell divisions.
And the sources even detail specific waves of this resetting, particularly for DNA methylation.
Yes, they do. Two major waves of global demethylation are described. The first happens very early, during that pre-implantation phase, around week 1 of embryonic development in humans. This affects all the cells of the nascent embryo, contributing to that initial clean slate or ground state. Then a second, equally significant wave of global demethylation occurs, but this one is much more specific. It happens only in the primordial germ cells, or PGCs, the cells that will eventually give rise to sperm and egg cells.
Ah, so resetting the germline.
Precisely. This wave in the PGCs reaches a minimum level of only about 5% CpG methylation around week 7 to 9 of gestation. This is crucial for erasing parental imprints and ensuring the germ cells are epigenetically reset for the next generation. After the embryo implants in the uterus, a process called de novo DNA methylation takes place. This is where new, specific epigenetic patterns are laid down across the genome, establishing the unique epigenomes from the different developing tissues and organs. However, there's a remarkable exception noted in the sources. Most CpG-rich promoters, these crucial regions right at the start of genes where transcription initiates, remarkably remain unmethylated throughout this entire dramatic process of embryogenesis.
Why is that?
It's a thought. This ensures that fundamental, essential housekeeping genes, which are needed in all cells, can always be expressed. Their promoters are kept open, largely unaffected by these large waves of methylation and demethylation. It's an incredibly precise, time-sensitive process that sculpts our initial cellular diversity and sets the stage for a lifetime of specialized cellular function.
And this precise orchestration continues, doesn't it? Once a cell differentiates and commits to a specific lineage, say, becoming a muscle cell or a brain cell, its epigenetic landscape becomes, you said, increasingly constrained. It locks into its fate.
Yes, exactly. It's a very elegant system, almost like Waddington's epigenetic landscape model, a ball rolling down a hill with branching valleys. As the ball differentiates, it chooses a specific path or valley, a lineage. As it proceeds down that valley, the walls get higher and narrower, representing increasing epigenetic restrictions. This makes it incredibly difficult under normal circumstances for the cell to suddenly switch to another path, transdifferentiate, or to roll back up the hill, dedifferentiate, to an earlier state. Our sources explain this beautifully. As cells mature and differentiate, their overall epigenetic landscape, and consequently their gene expression patterns, become progressively less similar to the initial, highly plastic ES cell from which they originated. They become more specialized. After a cell commits to a specific lineage, the repertoire of accessible regulatory regions, enhancers, and promoters expands specifically for the transcription factors unique to that lineage, allowing those key identity genes to be expressed. At the very same time, the accessibility for transcription factor binding sites associated with other lineages clearly decreases. They become silenced, often through repressive histone marks and DNA methylation. This creates what our sources refer to graphically as walls of valleys. These are effectively strong epigenetic barriers that prevent cells from inappropriately switching states or reverting to an earlier undifferentiated state under normal physiological conditions. This stability is absolutely essential for healthy tissue function and organismal integrity.
But this stability, these walls, can also become a vulnerability, right? And this has profound implications for disease, especially cancer.
It does. If chromatin homeostasis, that delicate balance of packaging, accessibility, marks, and boundaries, is disturbed, cells don't respond appropriately to signals. Their identity can become unstable. For instance, the sources suggest that overly restrictive chromatin networks, where those walls are perhaps too high and rigid, create epigenetic barriers that are inflexible and prevent all types of necessary cell state transitions.
So the cell gets stuck.
It might get stuck, yes. This could mean a cell can't adapt properly, it can't differentiate when needed, or it can't undergo necessary changes like senescence or apoptosis, which could contribute to disease or aging. Conversely, and perhaps more relevant to cancer, excessively permissive chromatin networks, where the barriers are too low or leaky, allow multiple inappropriate cell state transitions. The cell loses its identity.
Cellular chaos, as you said before.
Exactly. This can lead to dedifferentiation, where a specialized cell reverts to a more primitive, proliferative state, or even inappropriate lineage switching. Both of these deviations from the norm, whether the chromatin landscape becomes too rigid or too loose, are now understood to contribute significantly to tumorigenesis. Cancer is often characterized by this loss of normal cellular identity, uncontrolled cell proliferation, and a pathological de-differentiated state, all rooted in epigenetic dysregulation. So this truly suggests a delicate balance is needed in the epigenome. Too rigid or too loose, and fundamental cellular identity and function can unravel, leading directly to serious diseases like cancer.
That concept of these epigenetic walls guiding and constraining cell fate is just fascinating. And it leads quite naturally to another remarkable insight mentioned in the sources. It's not just our brains that remember things, our cells do too. This concept of epigenetic memory seems to extend far beyond just our neural networks.
That's absolutely right. The core idea here is that the epigenome, through the stability of its marks and structures, is literally able to memorize past events or exposures, influencing future cellular behavior. And this happens in basically every tissue or cell type throughout our body, not just neurons. So yes, of course, our neurons store individual memories, our experiences, enabling us to recall events from our childhood. That's cognitive memory, and epigenetics plays a role there too. But beyond the brain, our immune system also memorizes past encounters with pathogens, like viruses or bacteria. This is the basis of immunological memory, allowing for a much faster and more effective response upon re-exposure. This involves stable epigenetic changes in immune cells.
Like trained immunity.
Exactly. Concepts like trained immunity are heavily linked to epigenetic reprogramming of innate immune cells. And perhaps more subtly, our metabolic organs like skeletal muscle, adipose tissue fat, and the liver appear to remember long-term lifestyle choices related to diet and physical activity. These epigenetic memories can profoundly influence how we process nutrients long-term and contribute to our susceptibility to metabolic disorders like type 2 diabetes or obesity.
So what's the molecular basis for this? How does a cell remember using chromatin?
Well, on a molecular basis, this long-term cellular memory, which, much like our long-term cognitive memories, needs to persist through cell divisions, relies on the stability of the epigenome. Memories, whether cellular or cognitive, don't change the underlying DNA sequence. That's fixed. Instead, the epigenome provides a mechanism that is stable enough to store information over very long periods, potentially a lifetime, yet dynamic enough initially to be shaped by experience or signals. The actions of those chromatin modifiers and remodelers we discussed, particularly those within the powerful polycomb, PCG, and trithorax, TRG group complexes, are absolutely key here.
Polycomb and trithorax, the repressors and activators.
Broadly speaking, yes. Polycomb complexes are generally associated with maintaining stable gene silencing, like setting up H3K27E3, while trithorax complexes are involved in maintaining active gene expression states, often involving H3K4NE3. These complexes work antagonistically to establish and maintain long-term, mitotically heritable memory of silent and active gene expression states across hundreds, even thousands, of genomic regions. This means that as cells divide, their memory of which developmental genes or response genes should be on or off is faithfully passed down to daughter cells. It's generally thought that histone methylation marks, be relatively stable, primarily store these long-term decisions, like those defining cellular differentiation states or immunological memory. In contrast, histone acetylation marks tend to be much more dynamic, mediating shorter-term, more rapid responses to immediate stimuli. It's a highly sophisticated system for cellular recall, adaptation, and identity maintenance.
And this memory can even cross generations. That was a really mind-bending concept, mentioned transgenerational epigenetic inheritance.
Yes, that's arguably one of the most profound and perhaps controversial areas in epigenetics currently. What our sources detail is that changes in epigenomic patterns, which can accumulate over a lifetime due to environment and lifestyle, sometimes called epigenetic drifts, are how our somatic cells, our body cells, record this lifelong information or experience. These drifts can then be inherited by daughter cells when somatic cells proliferate, ensuring that the cell lineage maintains its acquired characteristics or adaptations. Now, what's potentially even more profound and critically important for understanding long-term health trends in genomic medicine is that in the case of the germ cells, the sperm and egg that transmit genetic and epigenetic information to offspring, these environmentally induced epigenetic drifts may, at least in part, escape that reprogramming we talked about earlier and be transferred to the next generation.
So things experienced by the parents could directly affect the child's epigenome.
That's the implication, yes. This phenomenon leads to the concept of transgenerational epigenetic inheritance. It implies that the lifestyle choices, like diet, stress, exercise, and environmental exposures like toxins or famine of parents and maybe even grandparents, could actually affect the health, development, and disease susceptibility of their offspring and potentially even grandchildren without any changes to the underlying DNA sequence itself. Imagine, as the sources suggest, a grandparent who experienced a period of severe famine. Some adaptive epigenetic marks related to metabolism or stress response established in their germline might potentially be passed down, perhaps influencing their grandchildren's risk of developing metabolic disorders like type 2 diabetes later in life, especially if their environment differs significantly.
Wow.
It's a truly remarkable illustration of how deeply intertwined our environment, our lifestyle, and our inherited biology truly are, extending far beyond what we traditionally consider Mendelian genetics. It adds a whole new layer to heritability.
So, if the epigenome is this dynamic, constantly being written and rewritten, if it's capable of memory but also susceptible to change from our environment and lifestyle, that means we might actually be able to influence it for the better, to actively reprogram it, maybe? This potential for reversibility seems absolutely huge for medicine, potentially opening up entirely new therapeutic paradigms, doesn't it?
It really does. That reversibility is the key concept driving much excitement in the field. Our sources make it abundantly clear. Most epigenomic modifications are indeed reversible, unlike DNA mutations, which are generally permanent. This inherent reversibility directly implies significant therapeutic potential. And that's why epigenomics is recognized as one of the most innovative and rapidly advancing research areas in modern biology and biomedicine. The molecular hallmarks of epigenetic control, those writers, readers, and erasers, are seen as prime targets for direct medical interventions and treatments. The promise of what are being called epidrugs, drugs that target these epigenetic mechanisms, has significantly increased the number of compounds now in preclinical or clinical trials across a range of diseases.
So we're moving into an era where we might not just treat symptoms or kill diseased cells, but actually try to reprogram cells back towards a healthy state.
That's the ultimate goal, yes. Or at least nudging them epigenetically in that direction, perhaps making them more susceptible to other therapies or restoring some normal function. It's a fundamentally different approach.
Okay, so given this incredible potential for reversibility, what kinds of drugs, these epidrugs, are actually being developed to leverage this and target the epigenome? What are the main strategies?
Well, epidrugs are typically a class of small molecule inhibitors designed to specifically target those chromatin-modifying enzymes we've been discussing all along. This includes targeting the writers and erasers.
Yeah.
Enzymes like DNA methyltransferases, DNMTs, histone acetyltransferases, HATs, histone deacetylases, HDACs, lysine methyltransferases, KMTs, or lysine demethylases, KDMs. Currently, oncology, the study and treatment of cancer, is by far the main focus area for clinical epigenetics. There are many compounds already in preclinical or clinical trials for various cancers. These drugs primarily work by inhibiting enzymes that are often found to be hyperactive due to gain-of-function mutations, or are simply overexpressed in specific cancer cells, driving the disease. But it's not just about targeting the writers and erasers anymore. There's a growing and really exciting interest in developing drugs that target the reader class of chromatin modifiers.
Targeting the readers, how would that work?
Well, reader proteins, like bromodomain proteins, which recognize acetylated lysines, or methyl-binding domain proteins, which recognize methylated DNA, are crucial for interpreting the epigenetic code and recruiting other factors. By blocking these readers, you can disrupt the downstream consequences of aberrant epigenetic marks in cancer, effectively cutting off the signaling pathway. BET bromodomain inhibitors are a key example currently in trials.
Can you give a specific example of an approved epidrug?
Sure. A powerful example mentioned in the sources relates to EZH2 inhibitors. Now, genes encoding epigenetic modifiers themselves, like KMTs and KDMs, are frequently identified as driver mutations in various cancers. They're not just passengers. They actively contribute to the uncontrolled growth. Inhibitors targeting specific KMTs and KDMs, like EZH2, promise to be potentially more specific and maybe less toxic than some of the older, broader-acting drugs like pan-HDAC inhibitors or early DNMT inhibitors, which could have more off-target effects. So EZH2 is the catalytic subunit of the polycomb repressive complex 2, PRC2, and it's responsible for placing that repressive H3K27Me3 mark. An EZH2 inhibitor called tazemetostat has already received accelerated approval from regulatory bodies like the FDA for treating specific cancers like epithelioid sarcoma and certain types of follicular lymphoma, particularly those with EZH2 mutations or overexpression.
And how does it work?
By inhibiting EZH2, the drug reduces the levels of those repressive H3K27We3 marks globally. but especially genes that were abnormally silenced by hyperactive EZH2 of the cancer cells. This leads to the upregulation or reactivation of tumor suppressor genes and other genes that inhibit cancer cell growth. It essentially reverses some of the aberrant epigenetic silencing driving the cancer. So it turns silenced good genes back on. In essence, yes. This offers a powerful new strategy to combat cancer, moving beyond traditional chemotherapy by directly targeting the malfunctioning epigenetic machinery itself. And while oncology is currently leading the charge, clinical epigenetics is rapidly expanding. There are promising trials exploring epidrugs for other medical areas, including inflammatory diseases, neurological disorders like Alzheimer's, and even infectious diseases, indicating a very broad therapeutic potential in the future.
This all sounds incredibly promising. I mean, the idea of truly reprogramming cells back to health, or at least nudging them significantly, it feels revolutionary compared to older approaches. But for a field this dynamic and complex, what are some of the key challenges or hurdles that researchers are facing, and what are the future directions?
Well, it's definitely not without its challenges. Our sources highlight some significant hurdles that researchers are actively working to overcome. For instance, as I mentioned, achieving specificity is a major issue. The catalytic domain, the working part, of many chromatin-modifying enzymes, especially within families like the KDMs on lysine demethylases or even HDACs, can be structurally very similar.
So it's hard to hit just one without hitting others.
Exactly. This makes it really challenging to design highly specific inhibitors that only target the particular enzyme isoform that's problematic in a disease without affecting other closely related family members that might be performing essential healthy functions elsewhere in the body. Such off-target effects could lead to unwanted side effects or toxicity. Also, we have historical lessons. We've seen that certain broader-acting drugs that impact the epigenome, like some older immunosuppressive agents, can unfortunately increase the risk of opportunistic infections or even the development of secondary cancers later on. This really underscores the critical need for developing next-generation epidrugs that are not just effective, but also highly specific and much less toxic, ensuring that we primarily target the disease cells or pathways while minimizing collateral damage.
But despite these hurdles, the potential you're describing seems immense and truly exciting. I mean, imagine treatments that don't just aim to destroy disease cells, which is the mainstay of much cancer therapy now, but could actually reprogram them back towards a healthy, functional state. Or perhaps just nudge their epigenetic landscape enough to make them susceptible to other, maybe less toxic, treatments like immunotherapy. This really feels like a move towards much more precise, truly personalized medicine, where we could potentially tailor treatments not just to an individual's unique inherited DNA sequence, but also to their dynamic acquired epigenome, which reflects their specific life history, exposures, and the current state of their disease. It's a whole new frontier for healthcare, isn't it? One that promises to reshape how we approach many chronic diseases.
Absolutely. And speaking of environment and lifestyle influencing that epigenome, we can't forget the direct role of what we eat. Our epigenome isn't just affected by internal cellular processes and inherited predispositions. What we choose to put into our bodies plays a huge direct role as well. This fascinating and rapidly growing field is called nutritional epigenetics.
Nutritional epigenetics, so food directly talking to our chromatin.
In a very real sense, yes. What nutritional epigenetics explores is precisely how various dietary compounds directly affect our epigenome and, consequently, gene expression patterns. It turns out that those very chromatin modifiers we've been discussing, the writers, readers, and erasers, they don't work in isolation. They often utilize key intermediary metabolites as essential co-substrates and air cofactors for their enzymatic activity. Think about things like acetyl-CoA, the source of acetyl groups for HATs, S-adenosylmethionine or SAM, the methyl donor for methyltransferases like DNMTs and KMTs, iketoglutarate, a cofactor for TET8 enzymes and KDMs, NAD+, crucial for sirtuin deacetylases, or ATP, needed for chromatin remodelers.
And those metabolites come from our food.
Directly. These critical metabolites are derived right from the breakdown of the carbohydrates, fats, and proteins we eat. This means our diet literally provides the essential building blocks and energy currency that the epigenetic machinery uses to make modifications to our DNA and histones. The metabolic state of the cell directly fuels epigenetic changes. What's perhaps even more specific and actionable is that secondary metabolites found abundantly in many healthy foods, fruits, vegetables, teas, spices, and herbs, can also directly influence epigenetic enzymes. Compounds like genistein from soy, resveratrol from grapes and red wine, curcumin from turmeric, EGCG from green tea, sulforaphane from broccoli, and various other polyphenols. These have been shown in numerous studies to directly modulate the activity of transcription factors and key chromatin modifiers like ATEs, HDs, DNMTs, and KMTs. So the components of your morning cup of green tea or the turmeric in your curry aren't just flavors or calories. They are potentially potent molecular signals interacting directly with your epigenome, subtly tuning gene expression.
That's a truly profound idea. Food isn't just fuel, it's literally information for our cells, shaping our epigenome. So how does our body actually sense these dietary signals, integrate them, and what are the long-term implications of this constant dialogue, especially when we think about aging and overall health span?
Our bodies have sophisticated mechanisms for this specific nutrition sensing pathways are constantly at work within our cells. Our sources highlight some key ones. These include well-known pathways like the insulin IGF signaling pathway and the mTOR pathway, mechanistic target of rapamycin. These are primarily sensing nutrient abundance, particularly glucose and amino acids. These actually tell our cells times are good, we have plenty of energy and building blocks, let's grow and proliferate. On the flip side, we have pathways involving enzymes like SIRTs, sirtuins, which are NAD plus dependent deacetylases, and AMPK, AMP-activated protein kinase. These act as sensors for low-energy states, signaling nutrient scarcity or increased energy demand like during fasting or exercise. They often trigger responses aimed at conservation and stress resistance.
So sensors for feast and sensors for famine, roughly speaking.
That's a good way to put it. These pathways work together in a complex, interconnected network to integrate the overall nutritional and energetic status of our cells and tissues. And importantly, they provide real-time feedback to our epigenome, influencing chromatin structure and gene expression patterns accordingly. This connection is highly relevant to our health span, that period of life spent in good health, free from chronic disease. For example, engaging in regular physical activity doesn't just burn calories or build muscle. Studies show it induces measurable, beneficial changes in skeletal muscle chromatin. Things like increased levels of activating marks like H3K36 acetylation and changes in the localization and activity of key histone deacetylases like HDAC4 and HDAC5. These epigenetic shifts, driven directly by the metabolic demands of exercise, are thought to directly promote healthy aging, improve insulin sensitivity, and enhance overall metabolic function.
So exercise literally rewrites parts of your muscle epigenome for the better.
It appears so. This powerfully reinforces the idea that our lifestyle choices, diet, exercise, stress management, mediated through these epigenetic changes, have a profound and direct impact on how long we live healthily and resiliently.
So connecting all these dots, this burgeoning field of nutritional epigenetics, where is it ultimately headed? What's the goal?
Well, the field ultimately aims to precisely describe and integrate these complex genome-wide interactions between our diet, our metabolism, and our gene expression patterns via the epigenome. The really exciting long-term goal is to move towards developing truly personalized nutrition strategies. Imagine tailored dietary plans for individuals based not just on their genetics, but on their unique epigenome, their metabolic status, and their specific health risks or goals. These personalized nutritional approaches could potentially serve as powerful early therapeutic interventions or even preventive measures for common, complex diseases with strong lifestyle components like type 2 diabetes, cardiovascular disease, or maybe even certain cancers. The ultimate vision, as some of our sources suggest, is perhaps to eventually dissolve the traditional sharp distinction we make between food and drugs, to move towards a much more individualized, proactive, and holistic approach to health, where what you eat and how you live is recognized as being as potent and potentially targeted as any conventional medication, working through these very epigenetic mechanisms we've discussed today.
Wow, what an incredible journey we've taken today through the world of chromatin. We've covered so much ground. From the fundamental organization of our DNA, precisely packaged into those intricate nucleosomes and then folded into topologically associated domains, TADES, to the incredibly dynamic interplay of those amazing molecular players, the writers, readers, and erasers that constantly sculpt and fine-tune our epigenome in every cell. We've seen how these complex layers are absolutely crucial for, well, pretty much everything, from the initial differentiation of that single fertilized egg into hundreds of specialized tissues, to how our cells develop these long-term memories of environmental exposures or developmental decisions, and even, critically, to the very onset and potential treatment of complex human diseases like cancer. This is truly the dynamic frontier of biology, isn't it?
It absolutely is. And this deep dive into chromatin remodeling and organization truly underscores, I think, that genomic medicine is far, far more than just sequencing DNA letters. That's just the starting point. It's really about understanding that profound dynamic regulatory layer of the epigenome, how it changes over time, how it remembers past events and lifestyle choices, how it contributes to disease when it goes wrong, and critically, how we can potentially learn to influence it therapeutically for better health outcomes. This new understanding offers a revolutionary dimension to diagnosis, to prognosis, and especially to the development of tailored therapeutic interventions. It moves us much closer to truly personalized medicine that considers not just your static genetic blueprint, but also the dynamic, ever-evolving instruction manual, the epigenome, that guides its expression throughout your entire life.
So here's a final thought to leave you with. If our lifestyle choices, our diet, our exercise, our stress levels, and our environment are constantly rewriting parts of the cellular instruction manual, our epigenome, shaping not just our own health, but potentially even influencing the health of future generations through those germline modifications. What responsibility do we have then, both individually and collectively, to actively choose behaviors and environments that shape an epigenome promoting resilience, vitality, and health throughout our lives and maybe beyond? What new frontiers will you explore in the realm of genomic medicine now that you've glimpsed this dynamic, intricate, and frankly awe-inspiring dance of chromatin?